We've learned about covalent bonds before, like the ones between the atoms of this molecule, thymidine phosphate. Covalent bonds are strong, meaning that it takes a lot of energy to break one, about 2 to 800 kilojoules per mole, and that they are about sharing electrons in the valence shell of two atoms, that is, the outer electron shell. Making and breaking these covalent bonds is the essence of chemistry and, of course, biochemistry. However, there are other attractive forces between molecules or parts of molecules that are of really great importance in biochemistry, the non-covalent bonds. These tend to be much weaker, most of them less than about 30 kilojoules per mole, and in one way or another they all come about because of unequal sharing of electrons between nuclei, which makes a covalent bond polarized and the molecule therein polar. This in turn has a knock-on effect on molecules nearby, and the consequence is a non-covalent interaction between two different molecules, intermolecular, or different parts of the same molecule, an intramolecular interaction. So while all the bonds you see in this molecule here, thymidine phosphate, a building block of DNA, are covalent bonds, thymidine forms non-covalent bonds when it's part of DNA. You can already see from this example that non-covalent bonds have a great role for intra- and intermolecular interactions in the cell. They also determine solubility in water. So what is the basis of bond polarity? It's all to do with the different electronegativities of the elements. The electronegativity is basically a measure for how greedy a nucleus is to draw electrons to itself. Fluorine is the greediest of all elements, but it doesn't play a big role in biochemistry. Important here is oxygen, which has a higher electronegativity than nitrogen, followed by carbon and sulfur, which are pretty much the same, and then hydrogen. What does that mean for biomolecules? It means that of the most typical bonds we encounter in biochemistry, only the carbon-hydrogen bond is essentially non-polar. The sulfur-hydrogen bond is moderately polar, but bonds between oxygen or nitrogen on the one side and carbon or hydrogen on the other side are all strongly polar. Polar bond has a partial ionic character, which means that the electrons tend to be more on the side of the atom with a higher electronegativity. The consequence is that all of the functional groups we've learned about before are polarized in this way. For example, the carbonyl group like this, the amino group like this, and the hydroxyl group like this. You get the drift. The uneven distribution of electrons means that we have a slight separation of charges. There's a partial negative charge with the oxygen here, and a partial positive charge with the hydrogen as well as the carbon. We indicate that with a small Greek letter delta. The uneven distribution of charge has consequences. It turns the molecule into an electrical dipole. We talk about the polarity of the molecule. Just on the side, a molecule is only a dipole if it has a net dipole moment, and that is a parameter indicated by these arrows pointing from positive to negative partial charge. As you can see, it depends on the geometry of the molecule. The polarity of molecules has a big impact on forces between them. The partial positive and negative charges attract each other. That means, for example, that more polar molecules hold on to each other more and have a higher boiling point than apolar molecules of the same size. Polarity also determines the solubility of the molecules in water. Polar molecules are much more water-soluble than apolar ones.